Dear students, the topic is activity of salivary amylase in relation to pH. In the last class, we discussed about the activity of salivary amylase in different temperatures and the calculation of temperature coefficient. Here we can see the activity of salivary amylase with different pH. Enzyme requires optimum pH for their activity. You know what is optimum pH? The pH at which enzyme acts normally is called as optimum pH. For salivary amylase, the optimum pH is between 6 and 8. That is 6 to 8 is the optimum pH for the activity of salivary amylase. Then what happens when the pH is decreased or increased? Enzyme will denatured. Enzyme will become denatured and the activity will be affected. Here we selected different pH like pH 4, you know, it is acidic, 7 is neutral and 9.2 is alkaline. These three different pH variation, how the activity or the rate of salivary amylase occurs. In this experiment, to determine the rate of activity of salivary amylase, or other name is ptyalin, on the digestion of starch, that is the substrate, with the effect of pH using Schooler's sugar titration method. In this method, the velocity of enzyme reaction is measured by determining the rate of disappearance of the substrate starch or the rate of formation of the end product. What is it? Sugar, that is maltose or glucose. Here in this method, the reagents used are the same that we used in the previous experiment. The calculation with the different pH is based on the comparison with Schooler's glucose conversion table. Have a look on detailed procedure of how to determine the rate of activity of salivary amylase with different pH. Before going to the procedure, we can see the preparation of reagents. First, enzyme amylase preparation. Rinse the mouth with water, collect the saliva in a beaker, dilute the saliva to a non-concentration that is 20% and filter through a glass wool. Buffer solution. Powder the BDH buffer tablets of pH 4, 7 and 9.2. Dissolve in 100 ml of distilled water separately. Substrate preparation. Dissolve 0.5 gram of starch powder in 200 ml of distilled water. Boil and cool down. Then filter through a glass wool. Sodium chloride solution. Dissolve 1 gram of sodium chloride in 100 ml of distilled water. Fillings A solution. Dissolve 34.65 grams of copper sulfate in 500 ml of distilled water. Fillings B. Dissolve 174 gram of sodium potassium tartrate and 125 gram of potassium hydroxide in 500 ml of distilled water. 30% potassium iodide solution. Dissolve 30 gram of potassium iodide in distilled water and made the solution up to 100 ml. 30% sulfuric acid. Add 30 ml of sulfuric acid carefully in 70 ml of cool distilled water. 0.1 normal sodium thiosulfate solution. Dissolve 25 gram of sodium thiosulfate in 800 ml distilled water. Here take 3 test tubes and mark them as A, B and C. In each test tube, add 3 ml of saliva plus 1 ml of substrate, 1 ml of sodium chloride and 1 ml of buffer solution. Buffer solution means, pH 4. In test tube A, add 1 ml of pH 4 buffer solution. In test tube B, pH 7 and in test tube C, pH 9.2. 1 ml buffer solution should be added. 3 test tubes should be incubated at room temperature perfectly for an hour. After that, take 10 ml of fillings A, 10 ml of fillings B, 5 ml of incubated mixture and 25 ml of distilled water in a Corning conical flask. Mix the contents well and boil exactly for 3 minutes first, then continue for another 2 minutes. Then cool the contents rapidly under running water until lukewarm. Add 10 ml of 30% potassium iodide and 10 ml of 30% sulfuric acid. Add 0.5 ml of starch as indicator. Blue, dark green or dark yellow color develops. Titrate with zero point 0.1 normal sodium thiosulfate taken in the burette. End point is disappearance of the color, or that is called achromic point. This table shows the summary of the experiment. At pH 4, the rate of enzyme activity is 1.39, in pH 7 1.29, and in pH 9.2 it is 1.36. Here we should calculate enzyme activity for 1 milligram of glucose formed in 60 minutes. For this, like that of the previous experiment, volume of sodium thiosulfate used should be noted. Milligram of glucose present in 3 and 4 ml, that we will get it from Scholar's glucose conversion table. Then after that, we can calculate enzyme activity for 1 milligram of glucose converted in 60 minutes. So in this way, for pH 7 and pH 9.2, it should be calculated. This is the same glucose conversion table used in the previous experiment. Dear students, I hope you can do this experiment in your lab. Thank you for watching. See you in next class with another topic.